Sige,
Ulit
sige,
from
sige.
the start.
And go.
Good day, everyone. We are the second group to conduct an experiment. The test was assigned to us is the Fehling's test. For us to further understand Mr. Herman von Fehling's test, may I have Mr. Gardose and Ms. Garcia.
So for our general objectives, by the end of our report, we should be able to define Fehling's test, discuss the Fehling's test using virtual experiment, present data gathered, and formulate a conclusion based from the experiment conducted.
Nakamute ka ata, Justine.
What is the Fehling's test? The Fehling's test is one of the most common tests used for the estimation or identification of reducing sugar and non-reducing sugars. This test is often used to distinguish between functional groups of ketones and carbohydrates that are water soluble.
Who invented the Fehling's test? Hermann von Fehling was a German chemist best known for developing Fehling's solution for sugar estimation.
Why is the Fehling's test important? The Fehling's test can be used to determine whether an aldehyde or ketone contains a carbonyl group. Aldehydes have a tendency to oxidize and produce a positive result. Ketones do not react apart from alpha hydroxyketones. The compound to be tested is added to the Fehling's solution and the mixture is heated. Initially, the solution exists in the form of two separate solutions which are labeled as Fehling's A and Fehling's B.
It's experiment time. So guys, are you ready? So the materials you'll need for our experiment is the glucose, lactose, sucrose, starch, Fehling's A and B reagent, dropper, test tubes, and beaker. So for our procedure, for our virtual lab to be presented by Alexander Tropa.
Mika, naka-share screen ka pa.
So now let us proceed to our virtual lab. So we will start with the glucose. As you can see here, you'll need to take 2 ml of Fehling solution A using a dropper. Then we are going to add it in our test tube where our 2 ml glucose is located. Right after that, we are going to add the Fehling solution B, same measurement as the Fehling solution A, 2 ml as well. Now, as you may have observed, the glucose inside this test tube turned into a deep blue color. Now we are going to get the test tube holder and remove the test tube from the rack and place it in a boiling water for a few minutes. Glucose is a reducing sugar, therefore it reduces the Cu2 ions in the test reagent. That is why it turned into a red precipitate of copper 1 oxide. Now moving on to sucrose. Same procedure that we did in our glucose solution, we will take 2 ml of Fehling solution A. Then we will add another 2 ml Fehling solution B to our 2 ml sucrose solution. Here, as you can see, that there are same measurements. Notice that the sucrose also turned into a deep blue color right after adding the Fehling solution A and B. Then we are going to proceed to get the test tube holder and remove the test tube from the rack and place it in a boiling water for a few minutes. As you may observe, the color remains the same. Therefore, our test for sucrose solution is negative, indicating that the sucrose is a non-reducing sugar, meaning carbohydrate that is not oxidized by a weak oxidizing agent. Now let us proceed to starch. Again, same process. We are going to take 2 ml of Fehling's solution A. And we are going to place it into the test tube containing the 2 ml suspension of starch. Now we're going to take another 2 ml of Fehling's solution B. Same procedure, we are going to add it into 2 ml solution of starch. Now you can see that it turned also into a deep blue color. Now we are going to place it into a boiling water for a few minutes. As you may have observed, the color remains the same, just like in our sucrose solution. It means that our test came out negative, indicating that the starch does not contain reducing sugar, meaning is any sugar that is capable of acting as a reducing agent.
Now, as you may have noticed that in our virtual lab, we only had 3 solutions. The solution lactose was not mentioned in our virtual lab, but if you will conduct the Fehling's test using the lactose solution and follow the procedure correctly, the result for lactose will be a positive Fehling's test because lactose has reducing properties. And now, for my own observation for the glucose and lactose, they both turned deep blue after the drops of Fehling's solution A and B. After boiling it for a few minutes, it turned red. Therefore, it means glucose and lactose both has a reducing sugar.
Based on my observation of the sample, sucrose and starch are the same. The starch and sucrose turn deep blue after the drops of Fehling's solution A and B. After boiling it for a few minutes, the color stay the same means it's negative. It indicates that starch and sucrose does not contain reducing sugar.
In conclusion, Fehling's test is used to distinguish between the presence of aldehydes and ketones in carbohydrates, as ketone sugars, except alpha hydroxyketone, do not react in this test. Always remember, the appearance of a reddish-brown precipitate indicates a positive result and the presence of reducing sugars, while the absence of the reddish precipitate indicates a negative result and lack of reducing sugars.
And we can also witness the Fehling's test in our labs, more specifically in the medical world, because Fehling's test is performed in medical facilities to detect the presence of glucose in urine. This helps to identify whether the patient has diabetes or not. So with that concludes our experiment. Thank you very much for listening.